Again, move on to the slides that Compo and Persner have shared as part of their book to the instructors. So we will look at how do we locate disease-causing mutations using these pattern matching algorithms, also known as combinatorial pattern matching. And we have already mentioned about this personal genomes, identifying different people's genomes and the mutations in their genomes, because it is incredibly useful in the setting where there are certain rare mutations which have very large phenotypic, very large observable changes, right? So phenotype is an observable behavior, very large effects in terms of disease manifestation, right? So Nicholas Walker is shown here is the first person to have the life saved by genome sequencing about 2010. Okay, so personal genomics can not only save the lives of people who are suffering from rare mutations, because then we will have a catalog of their mutations in everyone, and we can see which mutations are not causing any harm and which mutations are indeed causing harm or some particular disease, and we'll be able to come up with specific treatments for a subset of them. In addition to those clinical uses, it can also tell us what is the variation in our genomes across a human population, across different ethnicities, across different groups, right? And so that will also help us understand the genetic basis of diseases and from a research viewpoint. So having re-emphasized that motivation, let's just try to now formulate our computational problem that we have already formulated somewhat, but let's make it a bit more concrete, okay? So we have the reference genome, which is the database that is genome is used for comparison, okay? Database genome used for comparison. This is the 90s genome, very high quality genome. And then how can we assemble these individual genomes efficiently using the reference rather than starting from scratch, all right? And like I have said said before, we could use assembly. You can come up with multiple copies of the personal genome, right? And then we can shatter them into pieces. We can sequence the reads. So each of these pieces will give you reads, and we can assemble it with the overlapping reads using the Eulerian path problem, for instance, right? The problem is constructing a De Bruijn graph takes a lot of memory, right? And then a machine in a clinic that would collect and map reads in 10 minutes would be very nice, right? And not one that will sit and then go on doing computations for several hours or a day, right? So can we use the existing structure of the reference genome to help us sequence a patient's genome? Okay, that is what we would like to do, like I have mentioned before. And that is where the short read mapping problem comes in. Determine where each read has high similarity to the reference genome. So this is the reference genome and these are the reads. GA, GGA exactly matches here. CC, ACG matches here with one mutation or one mismatch. Here is TGAA with one deletion, right? But like I said, initially we are not going to worry about deletions too much, but let's just focus on these first two things, the exact match and inexact matches. So the simpler question is exact pattern matching, right? Where do the reads match the reference genome exactly without any mutation? So which is this first part, right? GA, GGA is matching exactly. If CC, ACG is not occurring exactly in any of the reference genome, we won't consider it. Similarly, TGAA may not also occur exactly. So then we will say that that read doesn't have any matching location in the reference genome, okay? But for GA, GGA, we will say that it will match at location 1, 2, 3, third location, okay? So the single pattern matching problem is a string pattern and a string genome, all positions in genome where pattern appears as a substring, okay? This could be one way of formulating a problem. If you have a single pattern and you want to find not just the first matching position in the genome, but all position where the pattern appears as a substring, okay? But you can also change it into the multiple pattern matching problem, which is the more clinically relevant problem that we have here, which is you have a collection of strings patterns and a string genome, and you want to find all positions in genome where a string from patterns appears as a substring, okay? So you want to find all matching positions in the genome for all patterns in the input set of patterns. This is still an exact pattern matching problem. We are not going to allow any mismatches or insertions or deletions. Okay. This is a brute force approach that that I already mentioned before, but showing here in terms of an illustration just to drive home the point. We can simply slide each pattern down the genome, right? So we are not going to do ACGTs here. It makes it easier to use normal English letters, but the ideas are the same. Even if this P, A, N, A, all these 26 English letters are changed to our DNA letters, DNA alphabet, ACGT, the idea will be the same, but it will make it easier to explain the algorithms and data structures today with normal English strings. So we'll just use a word like Panama bananas. Okay, that would be your genome. And your pattern is NANA, Nana. Okay. Where does Nana occur? So you can simply scan through this. So here it's not a match. So you just go to the next position. You compare AN is not a match. You go to the next. This is a match. This is a match. But this is not a match. So you go to the next position and you keep on going. ANN is not a match, and now you come to this position. You have N match, A match, N match, A match, right? So you have NA, which is matching, and then you have all the other locations which is not matching, right? So the location that it matches is NA, and that would be 1, 2, 3, 4, 5, 6, 7, 8, 9. So the 9th location in the genome is where Nana matches. And that was easy, like we had said before. It is so we would like to improve on this for it to have a better chance in the clinic. How will you do that? So brute force is too slow. The running time of the brute force is quadratic. Like I said, for a single pattern, it is order of genome times pattern. For multiple patterns, is order of genome times the size of all patterns put together. So patterns is the combined length of all of the patterns. Okay. If each pattern is 200 nucleotides, right, 200 letters long, and there are billions of patterns, this will be billion times 200. And the genome could be another billion. So this is just simply not going to make it to the clinic if it is such a long running time. Okay. Now, how can we improve this running time? Let's just explore certain ideas one after the other, and then eventually we are going to come up with some data structures called suffix trees and suffix arrays to address this problem in linear time. Okay, but let's just take one small step at a time. So these patterns all travel one at a time initially, right? So this was scanned, the pattern 2 was scanned, pattern 3 was scanned, pattern 4 was scanned, and pattern 5 was scanned. That's why you got this quadratic running time, right? But can we scan all of them together, right? Can we pack all the patterns into a common data structure and then scan that single data structure across the whole genome? Okay. And here the analogy is that instead of having everyone ride a car, we don't have that much space on the streets, right? So we use a public transportation. We just pack all the people who came into cars. All the patterns are packed onto a bus, and then we just run the bus once. Will it lead to any savings, right? And of course, we need to know what the metaphor for a bus is. What is the bus representing? A bus is representing something called a tree. Okay. The idea is to combine the reads into a graph. Each substring of the genome can match at most one read. Okay. So each read will correspond to a unique path through this graph. Okay. This will become clearer in an example. This graph is called a tree. Okay. Okay. This is just basically going to combine all the patterns into a single data structure, and this turns out to be a graph data structure, and this is called a tree. Okay. This looks like a tree, so maybe the tree and a tree, I mean, you can remember it that way as well. So it just has a similar sounding name. So if you have patterns such as banana, pan, nab, antenna, bandana, ananas, and nana, you are going to construct a data structure that will share most of the prefixes of these different patterns. Okay, so for you just go through all the patterns to construct this tree data structure. You have banana, so you just go ahead and construct a tree which is now a linear path that goes from all the way from banana, right? So BANA. So you construct a root and then several nodes, and then each edge is going to be labeled by one letter of your pattern. Okay. Now if you have something called a pan, you do the same thing again. So you have PAN, right, and AND. So all of these are just separate branches that are coming off the root. But once you see something that shares the same prefix, then you don't start a new branch, but you try to exploit sharing as much as possible. Okay. So AND doesn't have any sharing. NAB also doesn't have any sharing. All the first letters B, P, A, and N are not sharing, right? All of these letters are distinct letters. So each of them is going to have distinct branches. These are distinct letters. But the interesting thing comes from the next one, where there is an A, right? And A is going to share with this A over here, right? So then you can start this antenna, and in fact N is also going to be shared with this N here. Only at the third letter T and D differs, right? So then what we can do is the following. In this tri data structure, AN is going to be traversed from the root till here, and then you don't see a T, then only you start diverging and create a new branch here for antenna. Okay. I hope that is clear. And now you go on and then do bandana. Bandana also, ban is going to be this ban and this banana are shared, right? So when you have this bandana, from the root you see if there is any B leaving, so you have a B leaving, and then from there you see if it's a leaving, N is leaving, is there a D leaving? No, then bandana has to be some separate branch from the N. Okay, well, let's just see that. You have B, A, N, D has to be created a new branch, D, A, N, A. Okay. Similarly for ananas, you have A, N, you need to create a new branch, A, N, A, S. Okay. And nana finally, which is N, A, N, A. This is how a tri is formed. This is the metaphorical bus that we have mentioned in the previous slides. So all these patterns are packed into this tri data structure, and then you want to run the genome through this tri data structure, or this data structure through the genome. Okay. So you want to slide this tri down the genome. At each position you want to walk down the tri and see if you can reach a leaf by matching the symbols. Analogy is like bus stops, right? Let's just look at this. Panama bananas is our genome here, right? And then we are going to now see if at this position 1, if any of these patterns are going to be matching at position 1. And the way to check that is you just simply see every letter whether there is an outgoing edge labeled with that letter, right? So you have P, so you will take this path, you have A, you have this path, you have N, sorry, you have N, that is this path. So PAN is going to be matching the first position, right? So let's just do that. So you have P, you have A, you have N, so it's matching the first position. So you don't have to run all the other patterns here, because you have already detected that one pattern is matching here, right? And this would be the longest pattern that is matching at this particular position. Without exploring all the other patterns, we have just found the hit in the first position, right? Similarly, you now move the bus to the next location and you are going to check A and A and so on. So you have A, N, A, and then you have M, A, B, but then that is not going to happen because this is N. So you have hit a mismatch. So the second position doesn't match any pattern, right? Okay, so in the second position, there is no match because we haven't reached till the leaf, right? All of these patterns end at the leaf. So if you can come from the root all the way till the leaf, you know that you have reached a pattern. Otherwise, we have only reached a prefix of a pattern. So ANA is going to be prefix of one of the patterns, but it's not the full pattern. So you don't detect, you have not detected any pattern at the second position. So similarly, you can go to the third position. You have NA, and then you have none of these outgoing branches are matching. So there is no match at the third position in the genome also. Then you have A, no match at the fourth position. Then you have M. M is not even the first letter of any of our patterns, so there is no match at this position also. And now we have A, again no match. Now you have bananas. So this looks like there's going to be a match. We have BA, NA, NA. So that is the longest matching pattern there, right? And then you have ananas also going to match here. So another good match there. You have nanas. That nana is going to match there. You have ANA, S, but then that's not going to match, right? You have NAS not going to match, AS is not going to match, S is not going to match. Okay. So we have found all the locations where it does match, right? So especially we looked at this PAN was matching. This was PAN. We found banana was matching. We found ananas was matching. And so basically those are the matches. And this was just one scan through the genome. Okay. So what is the running time of the brute force? It was order of genome into the total size of all the patterns. The running time of the try matching is going to be the try construction. If you remember, we were going through every pattern one at a time and adding these different branches, right? So it is going to be linear in the size of all the patterns combined, so order of patterns. Pattern matching itself, we were scanning this bus from left to right, and each of those positions you need to start. The longest, for instance, was this one, right? Something like this, right? So you have to start from a root and then you'll have to go along one of these branches all the way down to a leaf. That is the worst case running time for any particular position. And so the worst case is going to be the branch with the longest length, and that is going to be the length of the largest pattern, right? And that is going to happen for every location in the genome. So then that is going to be order of genome into the longest pattern size. Okay. So we have obtained some improvement. Instead of running every pattern separately along and scanning along the genome, we just scan this try along the genome, and if the patterns are all of similar size, then only the size of the longest pattern matters, right? Or even if the patterns are of different sizes, only the size of the longest pattern will determine the worst case running time, right? So this looks like a good improvement, because in our case we have short reads. All the reads are more or less the same size, let's say 200 base pairs. So then you have the size of the genome, which is 3 billion, and so you have a 3 billion times 200 compared to 3 billion times 200 times another billion for the number of patterns, right? So there is this additional overhead of we have to read through all the patterns, but that is anyway we need to do, right? Because we need to input these things. So order of patterns is just linear in the size of the patterns, combined length of all the patterns. This is a billion into 200 base pairs. That is not bad, right? So this looks already like an improvement, but can we do better? Can we even remove this longest pattern from this genome? I mean this multiplication by the longest pattern. We'll try to do that, okay? But we may not see all the details, but we will give the main ideas, okay? So let's look at before we get to that, right? So let's also look at, we only looked at running time now, but also look at memory analysis, okay? Because we have not just a few patterns, we have billions of patterns, right? So what will be the space required to store this try? If it is a humongous thing, then improvement in running time for a huge blow up in space is not going to help us. Okay, so our patterns are 39 and our try has 30 edges. Okay, the worst case edges is going to be there is no sharing among all the patterns, right? In that case, all of the patterns is going to have like a separate branch starting from root all the way to the leaf. If you have sharing like it is shown here, then you will have fewer number of edges, right? And a fewer number of nodes in the tree. In any case, the worst case number of edges is the star pattern. From the root, all of the patterns are separate branches, and so the combined length of the patterns will be the number of edges in the tree. Okay.